This is TWIS. This Week in Science, episode number 863, recorded on Wednesday, February 16th, 2022. How to make a time crystal. Hey there, I'm Dr. Kiki, and tonight on the show, we will fill your heads with sex ed, optimism, and fatigue. But first, thanks to our amazing Patreon sponsors for their generous support of TWIS. You can become a part of the Patreon community at patreon.com/thisweekinscience.
Disclaimer, disclaimer, disclaimer. Attention spans are crashing harder than an Olympic figure skater on a bobsled run. Or so the popular belief seems to be, and in some ways it is true. Attention spans of most adults are likely much shorter than they used to be, for a very simple reason. We now have smart devices. In ancient times, before texting and smartphones, if people had a question, they had to either talk to somebody who might know the answer to that question or look it up in a book. So, say you have a question that comes up on Sunday. You're talking with a friend and you're trying to figure a thing out and, you can't figure out what the thing is, what the name of the person is, and you want to refer to. Who was that guy who invented that thing with the books? You might have known the name of that guy once, but now it escapes you, and so it is absolutely urgent that you find out who it was as quickly as possible. So, let's look it up in a book. Books, by the way, were like websites that had every single page printed out. These pages were kept in a brick-and-mortar repository called a library. They would have to physically visit during certain times of the day. And now it's too late. So, let's call a friend. Calling a friend meant that the friend had to be home to hear that you were calling them. Phone was usually attached to the wall of their actual domicile, usually in the kitchen or in the living room or somewhere like that. Some of the high-tech humans, back in ancient times, had a recording device that would allow you to leave a message if they weren't home. Then they would call you back, often the next day, but then you had to be home likewise, or have one of those recording devices, and the whole conversation would go on like this for a week. So Monday, you leave the message. Hey, Blair, it's Justin. Who's the guy who invented the thing with the books? Totally can't remember, and it's super urgent. Thanks, bye. Tuesday, you might get a message back. Hey, Justin, it's Blair. Do you mean libraries? That's Ben Franklin. Hope that helps. Bye. Wednesday, Blair would get the message again from you. Hey, Blair, Justin. No. The guy who invented books. Thanks, bye. And then Saturday, you'd run into Kiki at the farmer's market and ask her the same question in person. She would say, Gutenberg? And we'd go, it was on the tip of my tongue. Whatever that means. Then Saturday night, you get a message on the answering machine again from Blair saying, you mean Gutenberg? But you wouldn't call her back because you already got the answer. And then Sunday rolls around again. You see that buddy who you first had the conversation with, and as soon as you see him, you put out Gutenberg. But now it's lost all context and reference. So a week's worth of attention was required to facilitate that one piece of lost information. Whereas in today's world, you would Google it on your device, say, yeah, Gutenberg, that's who I meant, and then just move on to the next thing. So while our attention spans may be shorter, so is the speed of our informational resolutions, which is why we are able to bring you a week's worth of science in just 1 hour here on This Week in Science. Coming up next.
I've got the kind of mind that can't get enough. I want to learn everything. I want to fill it all up with new discoveries that happen every day of the week. There's only one place to go to find the knowledge I seek. I want to know what's happening. What's happening? What's happening this week in science? What's happening? What's happening? What's happening this week in science?
Good science to you, Kiki.
And a good science to you too, Justin, Blair, and everyone out there. Welcome to another episode of This Week in Science. We are so happy to be here to talk about science. All the fun news that caught our eye from the past week.
Yeah. Hey, Justin, next time you want to do a teleplay as your disclaimer and I have a part, you can totally send me the script and I will participate.
I absolutely would have. The only thing is I wrote it seconds before airtime.
Oh, okay.
So
hot off the presses.
I feel like this has history. Oh, everyone, thank you for joining us. we do have the science, and, we brought all the stuff in the scripts and out of the scripts. I have stories about time crystals in the wild, party fish, and old brain clutter. What do you have, Justin?
Let's see. What did I bring today? I have got microbiome and energy levels, how to live longer and eat less, a new life form discovered by a spacecraft, and just good news about HIV.
I hope it's really, really good news. Not just good news. We'll see. Okay, what's in the animal corner?
Oh my goodness. I have stabby birds and what was optimistic animals. So that's good. But I also brought a fun story.
Sex ed and optimistic animals. Things that go together. Oh, yes. And optimism and stabby birds obviously go together.
Yeah.
All right. We are all looking forward to these stories and more. There's so much to talk about in the hour.
An hour.
Is it longer now? I don't know how long this is.
150% of an hour.
Exactly. Yes. But if you are not yet subscribed to This Week in Science, you can find us all places that podcasts are found. Look for This Week in Science. We also stream weekly Wednesday evenings, 8 p.m. Pacific time on YouTube, Twitch, and Facebook. You can find us also as TWIS Science on Twitch, Twitter, and Instagram. But all this stuff, if you forget it, just look for us on our website, TWIS.org. All right, let's dive in. Science time. Jumping into the deep end of the pool, except the pool is like hard and it hurts when it's frozen because, water does that sometimes. It turns into a crystal. Don't
dive into a frozen pool.
Don't dive into a frozen pool. Just skate on it like those wonderful Olympians who are doing their beautiful routines this time of year. Okay. That was my weird intro to a story about time crystals.
Okay.
Okay. Is that a real thing or is that from like a movie?
Right. It sounds like it's just a concept from science fiction or fantasy.
Good device. Like,
time crystals.
What do we need to do? We got to go get the time crystals from the Crystal Palace to fuel the thing. It's a bunch of tasks. This whole movie is going to be a bunch of tasks.
It's going to be amazing. It'll be a hit. Totally. Yes. But time crystals are actually real. They were first suggested about, almost 100 years ago now, 75 at least. And physicists have been trying to figure out, hey, can we make time crystals? And finally, a few years back, yes, indeed, researchers were able to create time crystals, but in a very specific kind of superconductor quantum situation that it's only in the laboratory can be created and it's not very useful for real world applications. Like crystals are a particular regular patterned structure of atomic matter that it's like a lattice where the lattice has a predictable symmetry to it. Okay. And that is something that occurs in space. Now, a time crystal has the same kind of lattice-like symmetry within it that can be broken, only it is in time.
Still, I'm not getting a good idea what.
I know. Yeah, it breaks the brain a little bit. So, the researchers who just published a study about this have been working on creating time crystals that can be taken out of isolated situations, isolated conditions, and actually work in the real world. So in their work that they published in Nature Communications, they have created a very small system in which they have just a little square millimeter of space that's filled with gas. And this gas is magnesium fluoride, and there's a glass resonator within it. And it gets hit by 2 laser beams. And the 2 laser beams are tuned with temporal symmetry tuned to each other. And so the frequency of the frequency tones that these lasers are tuned to, they have occasional connection. And so it's this resonance that happens that blips up at random. It's not really random. It's a crystalline pattern in time, which is so interesting. But anyway, they use a resonator to achieve this connection that these lasers are able to create. And really, I mean, what we need to know is that a time crystal is something that is physical, yet at the same time is a useful concept. And it can be used now that we've got this ability to create it in the wild, out in the world, not in like sub-zero temperatures, not under specific pressures. This is just like out in the open. It can be created. Now it can be used for very specific uses like timekeeping, very accurate timekeeping, because of the resonance that occurs within the glass, within the laser, and the resonance between the two of them. The system as a whole is what's considered the crystal, and that can be put into quantum computers. It can be used in fusion to be able to accurately create the timing that's necessary to allow fusion to occur on a particular pace to maintain sustainable fusion. It can be used for even just instead of atomic clocks, we could use a time crystal potentially. So maybe one day our cell phones will have time crystals in them instead of just GPS units to connect us to satellites to figure out what time it is.
I have a time crystal watch. You mean a Timex? Yeah.
No, time crystal.
Basically the same technology.
Yeah.
I don't get it.
I don't get it. I'm with Blair. I'm not getting it.
Yeah, so it's a weird the weird concept is they're taking photons of light, so it's a photonic system, yet at the same time it's the concept of time. And so when you're thinking of something happening over time, it's an oscillating system that changes over time. And the fact that there is more than one atom present in the system allows for a bit of chaos in the way that the system interacts with itself. And so because of that chaos, it is able to create this crystal. And it's mind-bending, it's a difficult concept to get.
And I'm not getting it because I'm still getting a cartoon. Because then I'm like,
I still think of it as resonating. If
I run, I run over time. Am I a time runner? Well, technically, on some level, yes. But not in the way that would be interesting. So that doesn't mean the story's not interesting. It just means that I just don't get it.
Yeah.
I need. This is when an infographic. Can we
would be very useful.
Can we have a graphics department?
Can we get the time crystal infographic, please?
Can we at least get somebody to write some analogies that will help me?
Yeah.
So the IEEE, an author at IEEE who wrote an article about time crystals back in December, Charles Choi, who we know also from Live Science and other places, he's a great science writer. He says that a time crystal is a new kind of matter that bears an uncanny resemblance to a perpetual motion machine. Its parts can theoretically move in a repeating cycle without consuming energy for eternity, like a watch that runs forever without any batteries.
Watch.
Timex.
You were close.
I was very on point there.
I'm just grabbing at nouns now.
Well, because this is the thing. Like in anything, like in any system, all we have is a box of words, and we compare it with another box of words. If you look up some of those words, you get more words. So if it's a new thing, you don't have concepts behind. All you have is the words, and those words are related to other things. But if it's really a new thing that you haven't really conceptualized yet, it's just a bunch of words. That's always how it is.
Hello, I will be defending my thesis today. I have before you a bunch of words. Here we go.
Words.
Well, Kiki, I will say, sounds like something fun is coming with time crystals, and I'm excited to talk about it again later, and maybe I'll understand more of it next time.
That's exactly it. So hopefully we'll get people to talk about time crystals with us here on the show sometime this year so that we can learn more about them from people who work on the subject who maybe have the words.
We can make a 3-dimensional graphic that explains it, and in the graphic it could explain, but this is a 3-dimensional representation. Actually, this would be taking place in time.
You froze at a very opportune moment. Please don't edit that, is all I have to say.
It was perfect. Oh my goodness.
I love it.
Yes.
Okay, great. Time crystals. Got it. 100%.
Time crystals. They are not just science fiction or fantasy. They're not physical crystals, but they are new phases of matter.
They don't go in a warp core.
They do not go in a warp core, but they could help to make fusion possible.
So they could go in a warp core.
Or a lightsaber.
Okay.
Yeah, maybe I'm overestimating.
Kyber crystals?
Yeah. New technology, interesting technology. It's the exciting point about it is that the concept can now potentially be taken out of specialized laboratories with very specialized equipment and now potentially make it into other real world applications.
I'll have.
And that's exciting. Well, maybe not right away, but soon. All right. Justin, tell me, you have some good news?
Oh, yeah, just good news. The news segment that really tries its hardest to bring you just good news about a subject that is usually wrought with heart-wrenching, depressing reality, the kind that makes you want to stay in bed for days hiding under your covers in total darkness, wishing over and over again that the bad world would just go away. HIV edition.
Oh, no.
Just good news. A woman undergoing treatment for both HIV and leukemia has been cured by researchers at New York Presbyterian Weill Cornell Medical Center, or so it seems, 14 months after discontinuing antivirals. She is still testing negative for the HIV virus. Only 2 other people have ever been cured of HIV infection. Both were also suffering from leukemia. Those patients had a bone marrow transplant from another person who carried this rare genetic mutation that is known to block HIV infection. In this new case, the woman received cord blood from a person with the same genetic mutation as the donor, the bone marrow donor from the previous 2 cured patients. She was also given blood stem cells from her first-degree relative. The stem cells were given to bolster the woman's immune system, while the donated cord blood cells could become dominant in her system. Women continued to take antiviral drugs to keep the HIV at bay for 37 months after the procedure until 14 months ago, and she is now HIV-free. Why did it work? Well, they don't know.
Yeah.
The researchers think the cord blood cells might have the ability to adapt to the new environment and may contain stem cells that aid in the process. The patient was female and of mixed race, which is important because most past studies involving efforts to treat or cure HIV have involved white males. Researchers say that cord blood is much easier to obtain than stem cells used in bone marrow transplants, and the procedure was much gentler to the patient than a typical bone marrow transplant would be. Both of the patients that were cured of the HIV in the past, by the way, those were men. Both also suffered pretty severe side effects. I think mostly because bone marrow transplants are pretty invasive, extremely invasive.
It wasn't it wasn't just that the bone marrow transplants were invasive. They had immune reactions. So they had the host versus graft disease.
Correct. But the just mild symptoms and side effects in this woman. So she's well recovered really from the procedure. That's it. Just good news. Somebody's been cured of HIV and leukemia on the side. Not bad.
Amazing on both fronts.
Oh, God, wait. I just got the Just Good News, and it was just good news.
For real.
Yeah.
I know. I was waiting for the other shoe to drop.
There's no dropping. It's just another shoe. This is how he lures us in.
Oh no,
to a sense of
later.
complacency. And then he'll maybe do a few very nice, just good news stories, and then
wait. Let me finish that story.
No.
So that was.
Nope, you're good here.
No, that was the end. That was the end of the story. That was the end.
Good.
Yeah, that's amazing. I mean, we hope like this is she hasn't shown any signs of HIV for several months since
14,
the end of the treatment. So fingers
great.
crossed that it just stays that way, right? Let's hope it stays that way, but it looks good. Claire, is it time for some sex ed?
Yes. This is not only a portion of the show about sex ed, but also a really segment. New York University, a peer-reviewed publication, says federally funded sex education are linked to a decline in teen birth rates. Really? So there's been lots of studies before, but they were all correlative. So this was an attempt to create a more causative relationship between sex ed programs and birth rates. And so this was looking at a 20-year period. It was a long-term assessment of the impact of comprehensive sex education in reducing teen birth rates by county. So they were able to see that this comprehensive information on sex, contraceptive, and reproductive health had a greater impact than abstinence-only programs. Really? But also, this was specifically capturing people's age, their county of residence, and whether or not there were government-funded sex education programs provided to teens in their county. And so they found after the first year of this funding, teen pregnancy rates dropped 1.5%. By the fifth year, it dropped 7%, and it appeared to keep going. So this is something where, over time, it gets better and better. Who knows why? Maybe it's just because they get better at teaching it and better at distributing the information. It could be that the community accepts it better over time because it's expected. There could be any number of reasons why that is. But ultimately, sex education prevents teen pregnancy. Yes, go ahead.
Well, yeah, so there was the reverse of this, of course, the reverse study, the inverse study that took place. Yes. The state of Tennessee at one point abolished sex education, and then in the years to follow became the state with the highest rate of teen pregnancy, which. Really?
Yeah. So the second story about sex ed that I brought that I think it kind of further illustrates this point is about a meta-analysis of peer-reviewed publications looking at the STI and HIV risk reduction via sex education that focuses on sexual pleasure and desire. So this is, for any of us that grew up in an area where we went to public school and we had sex ed, that's not really part of the conversation. It's
not
the
at
nuts
all.
and bolts. Sometimes,
Yeah, nope.
I still went to school when they separated the boys and the girls, which is insane for so many reasons. But anyway, so not a lot of them talk about sexual desire or sexual pleasure. But this shows that can improve knowledge and attitudes around sex, shocking, as well as improved condom use compared to those that do not. That totally makes sense to me. If you're talking about the motivation behind sex, then it's a lot easier to talk about ways to prevent infection and also pregnancy in the context of when you actually might be in the heat of the moment and wanting to have sex. It makes way more sense to me. But so this is a meta-analysis of research literature from 2005 to 2020. They found that incorporating pleasure into programs can have positive effects on attitudes and safer sex behavior. And the recommendation is therefore to revisit sexual education and health intervention approaches that do not acknowledge sexual experiences can be pleasurable.
Yeah.
This is also like just thinking about, if you think about the whole idea of consent, and you think about being sexually healthy and not being emotionally damaged from early sexual experiences and all these types of things, I could totally see this helping with that on the whole. Because also, if you acknowledge sexual pleasure, I'm thinking back to the dolphins from a couple weeks ago. You acknowledge sexual pleasure on both sides. That can change the whole dynamic of how young adults approach sex for the first time.
Well, if you're not approaching it from a position of guilt, shame, fear, it changes everything. It changes the entire way that it is approached.
That sounds like it would ruin everything.
Thank you for bringing the love of love. All right, we have, let's see, more stories. Moving on from sex ed, let's move to fashion. Move to the left, fashion. Move to the right. So, have you ever kind of wondered, like, oh, I'm going to be going to New York. I wonder what the big fashion trends are, like in different areas of the town. Like maybe I need to pack certain clothes to fit in, or I'm going to move to a new city. And how do I know if I'm going to fit in? And what's the fashion there? And how does that all work? Or maybe you're just curious about how trends have changed, even within subcultures or neighborhoods within areas over years. Brooklyn has not always looked the same. That is true. People have changed their clothing trends. Well, to answer this question, researchers at the Cornell Ann S. Bowers College of Computing and Information Science have been working on an artificial intelligence underground map of fashion. And for this artificial intelligence underground map of fashion, they have an algorithm that can recognize similar clothing styles and map them from social media to particular locations within cities. What they're interested in it for is the potential that, it can be a useful tool for visitors to an area, for new residents. But realistically, it can also be really an amazing tool for anthropologists, for people who study humanity and the trends in humanity and the things that we're into over time. And if this kind of a tool, this kind of an AI algorithm can just start collecting information and save it for a few hundred years, anthropologists in the future are going to have, and digital archaeologists, they're going to have such an amazing treasure trove of information.
Boy. So my fashion choices are being judged everywhere I go, everywhere there's a camera.
Right. So when you're at an event or you're going to a certain place, or, you live, in a certain area. And so the algorithm can kind of figure out this is where you are most of the time. And it kind of will map you into the general trend. But instead of just looking at just a whole city, what they're looking at, they're making it neighborhood accurate. So they have 2 human-centered benchmark programs, Hood Maps and OpenStreetMap, and they're polling residents of cities in the study to be able to determine how accurate things are. The group's underground mapping, it better captured the sense of a neighborhood than existing methods. And so they're able to give people insider knowledge of a city. And they're calling it the underground mapping tool. Yeah, for cultural underground. So it's neighborhoods they're interested about, not actually like the parts of a city that work together to make up the whole. So, yeah.
So I can see how the fashion industry would want to use that. Definitely, I can see that would be sort of or
trend hunting, right? Like it's that's happening. I didn't know about that.
To interview people, but now they can just do that.
All right, let's talk about new things.
Oh goodness,
you have new things?
This is a big
trends?
one.
Yeah.
A novel life form was
That's
found
big.
by a spacecraft.
What? Where?
Life
In outer space?
On
life
Mars?
on another planet. That's what we're looking for, but it's not the most important criteria. In fact, finding life on Mars could be considered a disaster if the life we find is a little too related to life on Earth. If, say, we find signs of life on Mars, return that sample from Mars, from the Martian surface, back to Earth, and discover that the signal is actually coming from an organism. Then we attempt to sequence the life form just in case, and sure enough, it has DNA, just like on Earth. And oh gosh, by golly gee, what were the chances? Excitement would be high as the analysis of the organism's DNA is run through the public genomic database to discover it has a striking similarity to a specific strain of fungi found here on Earth, when it's actually named after a researcher who studied fungi for 18 years, whose university participated in building the very craft that discovered the sample on Mars.
Oh, really?
Oh,
really?
Yeah. So good at killing everything on those spaceships, but
So
Sounds
yeah,
like-
but thankfully,
so
That's
good,
amazing.
but never good enough.
thankfully that didn't happen because it was discovered by the spacecraft adjacent to it in the assembly facility.
Oh, not in space. Oh,
No, but it was a novel. It was a novel
Little
strain
contamination.
found in a clean room. They named it after a longtime Berkeley lab microbiologist, Thomas Trock. Trock, Berkeley lab affiliate scientist in the Climate and Ecosystem Science Division, decades of experience studying microbes in extreme environments, with a specialty of focus on studying those microbes that would show up in the clean rooms where they were assembling spacecraft. So this new fungal strain shows the ability to produce a biofilm. It means it can attach to surfaces and survive cleaning protocols in the assembly facility where it was discovered. Strain's ability to survive in this low nutrient environment is of concern because these clean room facilities comply with NASA's robotic missions requirements, and this is something that's escaping the protocols. Getting around it, getting by it. So kind of fun. A scientist who's worked for decades on this, who's not, I think he's retired, got this named after him, which is a great honor to have a fun guy.
huge offer.
named
He's a
after-
fun guy for sure.
Yeah.
Yeah.
But it also it presents once again that issue of, if we go there and if we discover life, how do we know we didn't bring it? And Blair, your point, yeah, what if it escapes and manages to subsist upon Mars and propagate, and then that's the life on Mars? And then in the back of my mind, it's like, why not? What if the things go hell in a handbasket here on Earth, and we never do colonize? There is no Marsifest destiny. We don't get humans there, and the space program ends, and we all devolve, and nothing ever had. That was the height of humanity. Hey, at least life has another shot if we leave some trace of it behind on another. So there's part of that in the back of my head. It's like, if you found something that can survive a hostile environment, let it go.
Life finds a way.
But not
for
yet,
sure.
not yet, because we still need to do the initial investigation to determine that there is no life before we start polluting it.
But you don't want to land on Mars and have a nice squishy landing because it's just covered in a thick layer of mold.
Okay, so something like this that is existing in a clean room, it is an extremophile. So
Mhm.
if it can if it can exist through the harsh environment of space, through launch, through entry, through the atmosphere of another planet, to get to the surface of another planet, if it's extreme enough, what is that going to happen? Is that possibly going to be? Is there going to be the contamination? And if there is, we already have contaminated Mars.
Quite
Just bring
possibly.
some tardigrades and be done with it.
And
I mean,
I
we started
think-
throwing spacecraft at other rocks in this solar system before we were
Yeah.
really thinking about this cross-pollination
Mhm.
idea.
Really good point. Really good point.
And I think there's some admission that that's going to happen.
Yes.
I think the fear is that it interrupts the mission of sussing out life. I mean, the scenario I presented, where you go up, you get the signal because they're not in that,
Yeah.
being able to do the full spectrum of analysis, perhaps. But they get a signal that something organic is there, and then they take all the trouble to bring it home again, and it turns out it's the thing we brought there in the first place. Enormous waste of time, effort, and money. So we don't want anything, we don't want any noise in the system while we're out there doing our search.
While we're trying to figure out a new planet, we don't want noise in the system. Do no harm in that sense. But then if we want to colonize it, do all the harm we want. It's like...
Yeah.
Then the pendulum shifts.
Well, then it's not, if we can rule out life. I mean, it's almost in a sense to the progression of things. It might even be easier if we don't find life. Then we can just go there and colonize, not give it a second thought. That might actually be the easier way. If we do find life, then we have to really hand wrangle and struggle for a little bit about how long do we spend studying the novel native organisms.
It depends on how much trouble we're in here on our planet.
Yeah, that's always a fact.
Yeah. So many interesting questions. That is really cool. And meanwhile, on our own planet, we are just makety uppeting all sorts of little organisms. So researchers at Harvard have their School of Engineering are making fish, little teeny tiny fish based on zebrafish, but they're making them out of human heart cells, human cardiac muscle cells. Yes, they have created these little biohybrid, I shouldn't say biodynamic because that's like a word that's used in farming, which we're not talking about. Biohybrid fish. They're a little bit machine, a little bit human cardiac muscle cells. And the muscle cells, they have a pacemaker that they put in that gets it all going. And then the muscle cells act on the fact that one side is stimulated to contract. And while that side is contracting, the other side is stretched, which makes it contract, which stretches the other side, which causes an oscillation back and forth. And why on earth would we be using cardiac myocytes to create little fish? Like, why would
that's
we
what
be
I was
doing
gonna ask.
that? So what researchers are attempting to do in this effort to make these little heart fish is actually learn how to build artificial hearts. And so they've been building different structures for many years out of heart cells from different species. And they've finally gotten to the point where they're like, all right, we're using human cells to do this so we really understand how the cardiac myocytes work together. And so by creating a system, the researcher said, he's like, look, you can create a lump of cells in a dish and call it you can call it what are they calling those things? Those balls of cells that are organoid. You can call it a cardiac organoid, but it's still not a cardiac system. It's really not the kind of system that's doing what a heart does. It doesn't have the same kind of thing moving it isn't working the same way. And so they're learning how the contraction and activation of these cardiac myocytes works to see how they could potentially integrate it into an artificial heart system. One that would potentially last the lifetime of an artificial heart recipient. And that is the situation here. So these biohybrid cardiac fish, once they get going, They take a little while to get going, and they last for about 100 days, and they actually get stronger from the minute they get activated to the first couple of weeks. They get stronger and stronger and stronger, and then they just keep going. They keep fishing, fish. They keep swimming around. Their movement is based on the movement of zebrafish, and hopefully one day this will allow researchers to understand how to build a heart.
That is the most ridiculously brilliant.
Right?
It's
so
both. You're like,
cool.
that's just so ridiculous that you made a little fish swim thing. But then you can just like basically put them in the tank and see how long they swim, and you can determine how robust your gosh,
how robust the system is, and it's
brilliant.
the muscle working together. And the researchers were saying that now they can investigate mechanoelectrical signaling as a therapeutic target of heart rhythm management, and for understanding pathophysiology and sinoatrial node dysfunctions and cardiac arrhythmias. These little fish can potentially help solve a whole bunch of human cardiac problems.
What a fantastic
So
cool.
So the 100 days, is that because the pacemaker runs out after 100 days?
Right. Maybe that's just how long the experiment went before they published it, and they'll keep going. Yeah.
Here's where I'm getting confused. They don't have a digestive system. It didn't have a battery.
Right.
I'm assuming
they're in a
bath. Liquids that,
right.
Yeah, it's going to be in a bath of fluid.
Nutrient-rich, fluidy thing.
Right. But
not unlike what your head will one day float in.
Yeah. Hopefully for more than 100 days. Exactly. But so that's what I'm thinking is that something that lasts for 100 days in a nice nutrient-rich bath would do much better in a closed system with,
getting fed.
A metabolism. Right. Because it all depends on what broke down after that 100 days. If the muscle fibers are breaking down, if it pooped out.
Well, it's right.
Or and this point, even if anything at all, like it might just been, we're going to run this for 100 days.
Yeah.
And
and it could be, the system doesn't have all the stuff that would necessarily be there to fix problems. And we do know that cardiac myocytes, once they get broken, they can't fix themselves. They're not good at doing that. That's why we have a whole bunch of heart problems. But what they're learning right now is very, very valuable.
Yeah.
From these little teeny tiny fish. Oh, and Blair, this is a very interesting point. The biohybrid fish reached speeds and swimming efficacy that was similar to zebrafish in the wild.
Somebody at this facility, this team, somebody is too smart to just do one project. Like,
yes, I was thinking the same thing.
So I was just like, I'm going to engineer this. I'm going to make fish out of it. And I'm going to time them, and I'm going to make them a good model of a fish just on the side. Mm-hmm. On the side, because I can't just do the one thing.
You could just have muscle fiber that twitched.
That's it.
It'd be like, I did it. It's a heart. You can call
it
keep time. No, you went way further.
Yeah.
For time trials.
Fish. What if this heart muscle was a fish?
Cardiac fish races. Insanely awesome. I think this is my favorite story of the year.
This is someone who was rejected from ichthyology and was pushed into cardiology and was like, you know what, though? The fish are where my heart is.
This is one of those that I know we cured HIV at the beginning of the program, but this is one that I think we need to keep for the end of the year. Is like one of my top stories for the
all right.
End of the year.
We
HIV thing's very impressive.
We'll put it on the list.
And then was the driving fish, was that this year or last year?
That was this year also.
Yeah, the fish are killing it.
Fish. Oh, is 2022 the year of the fish?
It's the year of the fish.
I thought it was the year of the tiger. Come on.
Well,
tiger gotta eat.
This is This Week in Science. This is the day, the week, the episode of This Week in Science that you've chosen to listen to. Thank you so much for joining us today. If you are interested in joining us in the future, just know that we're here every single week. Please share us with a friend. All right, let's come back. I really did not want to bring any COVID news tonight. Justin, can you tell us what's going on in Denmark?
So Denmark opened up completely. No mask. Yeah, they still have testing, but if you test positive, you can basically still go out in public. They even getting rid of quarantining and everything. All out the window. It's endemic. We're going to roll with it. They started this a little while back, and cases and deaths have surged.
Mm-hmm. Surprise.
Okay. Yeah, surprise. Now, people are looking at them doing this in the United States. I want to point out something that is important. Denmark has, of the people who are considered vulnerable, most vulnerable, the elderly, the immunocompromised, they have 95% have been vaccinated. 90% of those have been vaccinated to the point of the booster. Okay. This is a country with universal health care. Where everyone goes to the doctor without thinking twice about it if they need to, because they're not going to get hit with a life-hemorrhaging bill that ruins their credit and their ability to progress within a capitalistic society. Those are big differences. The United States had recently the weekly total of 2,000 deaths a day, which, let me see what it is. If I put in the thing, it is okay. 365 times 2,000, the current rate. That's before everything everywhere is opened up, by the way.
Yeah.
That is 730,000 dead people in 1 year. And that's with the current level of masking. You get rid of that and you get a surge and oh my, I don't have a scientific on my, don't do it.
I read an article this week about endemic delusion.
Yes. Yeah.
So there's this idea. It's been sold to kind of people through, I don't know, the media or something. I don't know. But this idea, it's like if we all just give up, like it'll be fine. Like it's if you were gonna get it, you got it. It's done.
And in that article that you read, there's it's actually a script researcher who is actually from Denmark, who's like, I do not agree at all with what's going on in the homeland. And the people who are coming out in favor of it tend to be politicians, as is the same case in the United States. You're not getting a lot of people who study this sort of thing coming out and saying this is a great idea. You're getting people who want the political will.
And on that note, everyone, we will end our COVID segment. This is This Week in Science, and we hope that you are safe and healthy and happy. And we thank you for joining us for another episode of our Science News podcast. If you're enjoying the show, please head over to TWIS.org and click on our Patreon link, where you can choose your level of support to help support this show in an ongoing fashion. We do give you this show. We bring it to you, produce it every week for your enjoyment. We'll bring you the news. And if you support us at $10 or more per month, that's less than a cup of coffee a week, probably. $10 a month, and we will thank you by name at the end of the show. Because really, we can't do this without you. Thank you so much for your support. All right, let's come on back now. You hear? For a little section of the show with a few, I don't know, it's noisy. There's a bunch of animals in it. There's also knives this week. I don't know. It's Blair's Animal Corner.
With Blair.
She loves our creatures, great and small. Biped, millipede, no pet at all. If you want to hear about animals, she's your girl. Except for giant pandas and squirrels. Those animals.
What you got, Blair?
I have peacocks.
Oh dear, they're loud.
Yes. Yes. They are famous not just for being loud, but also for killing and eating snakes.
Ooh,
Wait, what?
Yeah.
Peacocks.
They're famous for killing snakes. And so
no idea.
yes. And so
Yes.
the Muzaffar Ali Khan devoted his PhD to investigating exactly how peacocks can kill venomous snakes. In Pakistan, where he grew up, he actually had many neighbors who kept peacocks, and so they kept them. One of the reasons was to kill snakes. And so, under the supervision of Michael Richardson at the Institute of Biology, Leiden, they analyzed the molecular resistance against cobra venom in various birds and mammals and found differences. What kind of differences, you ask? I'm so glad that you asked. They looked at several mammals that eat snakes first. They looked at mongooses, the European hedgehog, the honey badger, and determined that only mammals that shared territory with snakes had evolved some sort of resistance. Makes sense. The resistance made the snake venom less potent in their system by making the toxins unable to bind to the target in their body. But what's specifically interesting about this, they're resistant to venom. That makes sense. They eat venomous things. But what was interesting was that they found in the DNA that they were not the same in all the animals. The DNA markers that caused this adaptation were not consistent, which means that the different animals evolved the resistance each time independently. There was not a common ancestor that already had resistance. Which brings us to birds. Peacocks, hawks, eagles. One of my favorites, the secretary bird. They are all known for killing snakes. But when they looked at the DNA, none of them were even slightly resistant to venom. There were no genetic markers towards venom resistance.
Not at all.
No.
So if they get bitten by a snake, lights out.
You want to know their theory of how they survive these venomous snakes?
Feathers are actually fang-proof?
No, it's their stabbiness.
Okay.
It's the stabbiness. So birds, all of these birds, yes, of the birds, they're all very, very good at attacking snakes. They are fast. They have excellent vision. They have heavily protected scaly legs. They're very smart. They're very quick. They're very agile. And so it seems like there's no selective pressure for venom resistance. They're just too darn good at killing snakes.
They don't need resistance because resistance is futile.
Yes, there you go. I guess resistance is redundant in this case. But they also use feathers to distract the snakes. They know exactly where to peck at them, away from the fangs, back of the neck. So all these things mean that this is all still a theory, but this is the theory of why they are able to kill and eat venomous snakes. So a couple things about this. So first of all, that means there is not some common vertebrate ancestor with venom resistance, which would have been wild to figure out. I would have guessed it would be a long shot, but
I'm still not convinced that that's not the case. But
it would have to be
not
venom
a common
resistance,
one.
like back in the dinosaurs.
Yeah, no, I mean that's kind of almost what I mean, though.
Yeah.
Like, I mean, because it's odd that they had come up with resistance in different ways, which tells me it's not. It looks like a convergent thing, but I think you also have to have maybe building blocks for even if they come across differently. I don't know. That flexibility, that plasticity to come up with venom resistance that's different in different ways still kind of tells me that maybe there is an architecture for it somewhere in the way back, even if it's not just resurfacing or consistently. But yeah.
So you have to keep in mind too, though, peacocks are from India, secretary birds are from Africa, eagles and hawks, Europe, the Americas. So they all kind of live in different places. So if they are, you also have to think about the timeline of the specific venom that they're going
because not all
after
venom
too,
is the
right?
Same.
Not all venom is the same. In fact, in a study many years ago, I reported on if you can identify even like down to a few square miles of where a venomous snake is found, that can help you develop a better anti-venom. So in Israel, where there were many venomous snakes, a bunch of types of rattlesnakes and vipers and stuff, they would say if you get bit by a snake, try really hard to recover that snake and specifically take note of exactly where you were bit geographically, because that will impact the anti-venom that you use. So if venom is so specific to a species, to a location, I don't think it's that surprising that the method of resistance would be different.
Okay. But then the whatever architecture was there that then applies that resistance is sort of where I'm getting at. Sounds like that's a thing in common. But then it's
but that's the thing is it's not necessarily in common. It's going to have to do with a very specific location with you're an animal in a particular space and there's a particular venomous creature that you have to be resistant to.
Correct.
Versus like you're a bird who flies all over the place and you like to eat snakes.
Right.
Yeah, but that's my point is that it is emerging differently because the venoms are different. Like but you
Justin,
have to take an eagle and put it in India.
The main piece of this study, though, was looking at DNA.
Yeah,
and the fact that the location in the DNA, the specific DNA responsible for resistance, was not at all similar between the Asian mongoose, the European hedgehog, and the African honey badger, means that they do not have the same evolutionary origin.
Disparate, convergent affect. Yeah. Okay.
So
it's convergent in the fact that it's resistance to venomous to venom, but it is completely independent in the fact that it is totally different DNA mechanism.
That's what makes it convergent. Yes.
Yes. But it's also a really good example of how sometimes a behavioral tactic can be effective enough that there's no need for a genetic physiological change, which I think is also the other piece of this, that these birds figured out, and this could be convergent or it could have a common ancestor. There could be an entire bird ancestor that has passed through genes that show it how to kill snakes with it. But ultimately, the behavior was successful enough that there was no selective pressure to create venom resistance, which is very cool. A study from Ben Gurion University in the Negev, Israel, and that is in cooperation with the Department of Wildland Resources, wanted to look at optimistic or pessimistic thinking in wild animals and how that impacts their behavior, and also how it impacts their success, actually. And so they developed a computer model for animal foraging that considered valence-dependent optimism bias, which is a lopsided learning process in which information about bad outcomes is discounted or ignored. They showed that when
gambling's all about.
Yeah. It's gamblers, the ultimate optimists, according to this study. When faced with decisions, foraging animals that gave mental weight to positive outcomes had an on-the-ground caloric advantage. More optimistic animals did a better job. They survived better. They got more food. They did better, generally speaking, in the game of survival. In this simulation, the fictional animal forager was equipped with partial knowledge of average food quality and travel time to new food sources. They made ongoing decisions about whether to stay where they were or to return to previously explored areas or to go to new ones. So yes, this is gambling for sure. Every time the forager moved to a new patch, it learned about that area, it added that information to their mental library, and then they moved on. So optimism optimistic decision making led to healthier circumstances and better access to food overall. Even when it didn't reap food rewards in the short term, rapid learning was beneficial because if you continued to look further, you'd find more good stuff. And so animal mortality is driven by starvation, generally speaking, rather than predation. So access to food is ultimately, specifically for the animals that they study, these kind of grazing animals, foraging animals, that was the ultimate decider in success or failure, is were you able to get access to food? And so animals that used extremely optimistic strategies did better. Ones that used extremely pessimistic strategies tended to die of starvation at a younger age than the other individuals in the sample or in the computer program. Yeah, look at that happy deer. I know it's not actually happy, but it looks like a cartoon. It looks so happy. But
what
so
kind of grass are you eating?
Very happy grass. So,
go ahead.
Yeah. So, what is it really that? How can I have optimistic foraging strategies and not starve?
You know what this reminded me of? The sunk
What do I do?
cost fallacy.
Yes.
And it's kind of the opposite of that. So if you are stuck in a situation where you're not getting what you need, it is beneficial to be optimistic and take the plunge. It's it's like in job hunting. You on average, if you get a new job, even if it's a quote unquote lateral move, you make, I think it's 15% more every time that you leave your job. So it's a very well kept secret out there. It's true. So that means that taking the plunge, being optimistic, jumping to something new is beneficial to us as well. So that's actually a really good example of this theory in practice. But it takes optimistic thinking to think, okay, I'm going to fit in my new workplace. I'm going to like my new job. My boss isn't going to be terrible. Like all of these things, you really have to be optimistic to want to take the
try something new.
Yeah, because there also is a chance you're going to go and it's going to suck. And if that new job is bad, then you have to try again. That's ultimately what this strategy means, right? Is keep trying the new things until you find good stuff. As you could apply this to dating as well. If you're in the dating pool, right?
It's good to know though that like this comes from very solid ecological behavioral principles where this is animals making these choices survive versus animals that don't. They're gonna die. They're gonna die of starvation.
So what this model is telling us is you want to balance your known territory, whatever metaphor you want to apply that to. Balance your known territory with the time and energy devoted to exploring new resources. So you want to devote time to the things that you know are beneficial, but make sure you set aside time to try new things because you will find something beneficial to you if you keep looking. So it's what the researchers say, because it's so funny, is optimism is a mental glitch that offers an evolutionary advantage when dealing with limited information, spurring exploration and allowing for adaptations to changing environments.
Next time I see somebody who's being unreasonably optimistic, I'm going to tell them to put their mental glitch away.
No, I won't. This mental glitch. It's gonna be forever. So anyway, it's very interesting to consider when you apply it to humans. That's not what the study was about. But it also is really important to know when studying ecology and working in animal conservation. If you know the strategies that will be most effective, then you can help kind of predict where the winners of a population will go and how they will act. And so if you know that exploration is beneficial and you build a new shopping mall next to a wildlife refuge and there's more car strikes in the parking lot of that new shopping mall. I'm just making this up. But ultimately, if you do that, then you know that you are actually probably weeding out the most optimistic individuals in a population, and that could have a negative impact on overall population health.
Oh, that's a Oh gosh, that's a
huge
great insight.
implication. Yeah.
So yeah, so there's a lot that you can kind of apply with this information based on a computer model. But yeah, optimism.
Interesting.
It could be a good thing.
Very big insights for optimism. All right, Justin, I'm optimistic that you can tell us something energetic. Keep us from being fatigued, maybe?
Oh, yeah, that's a good intro to the next story that I've got. Clarkson University researchers found a correlative link between a small sample size of humans' energy levels and their gut microbiome. Associate Professor of Physical Therapy Ali Bouliani and his team searched the microbiome for associations between microflora and self-reported personality traits or mood ratings of mental energy, mental fatigue, physical energy, and physical fatigue. They had they had 20 participants assessed by survey. The gut microbiome and metabolome, which is the actual enzymes and what have you, molecules that are being produced by the gut microbiome. They were determined. There was RNA analysis as well as untargeted metabolomic analysis. Found some interesting things in here. Bacteroidetes, 45%, the most prominent phyla, was only negatively correlated with physical fatigue. So I guess that means if it wasn't there, you would see physical fatigue more highly rated. Second most predominant was butyrate producing phyla Firmicutes, and it has the traits. That one is kind of so bacteria anaerotipes. I'm very slow at pronunciating, but it was positively correlated with mental energy and negatively with mental fatigue and physical fatigue, respectively. So that's kind of an interesting one. Now you got this one that is there when people have mental energy and is sort of disappearing when they have mental fatigue, report physical fatigue. Diet sort of influences this too, because they're also tracking these people's diet. One food group in particular, processed meat, was correlated with all four of the moods positively with mental fatigue and physical fatigue, negatively with mental energy and physical energy. And that's the one that had the Firmicutes genus Hodamania. That was correlated with the processed meats. It was the only one that had just very specifically correlated. So it's sort of interesting because I've been mocking nutritionists, not mocking them wholeheartedly. Like,
we know the microbiome is influenced by diet and fiber, and there's all sorts of interactions.
Of course,
there.
But I've been mocking the nutritionists because they've written all these books. You could put a whole bookcase together of books written on nutrition before they knew anything about the microbiome and how it interacts.
Right. Yeah.
And so then you're like, okay, throw all that knowledge away and start over. But you can actually build off because now you're seeing
this is why
foods correlated to microflora in some cases. Now, the rest of them, they weren't, but this is one specific one that they found was correlated in the sample. But it's really interesting because this is starting to get into that field of what is a healthy, productive, or hacked microbiome. Now, you might hack it so you've got mental energy and you don't have any physical fatigue and you're going. And then it turns out that leads to some awful disease down the road that we don't even know about. But so it's a small sample size. They only had 20 individuals in this, but they are now seeking funding to do much larger, more robust study because the initial correlations, and this is a follow-up to other studies that this group has done. So that it looks like they're kind of on the right track there.
How do the microbes play into it? And I think that's a really interesting question.
And this is why this study, I think, was also very interesting. And again, like you're saying, the bigger study, when they get more data, is actually going to be probably more revealing and more interesting. is that you're starting here with a correlative, and this is when correlative science is not bad.
No, it gives you a place to start.
Definitives. It's giving you a starting point.
Place.
But if you do, then okay, so then you've narrowed it down to like, say it tracks and processed meat stays tracked to this one microbe that the gut microbe that tends to flourish. Now you can study that one gut microbe and see, okay, what are interactions? What molecules is it producing? How do those molecules, once produced, interact with the larger system? And now you're starting to get mechanism, the causal things, and then you can actually, like, if you need a drug treatment or a drug blocker or just a dietary recommendation that's actually backed up all the way through so it leads to all these things. You're right. This is but the other thing you could do is just stop eating.
No, you can't.
Just stop eating. Turns out,
Okay.
people who eat 14% more have reduced healthy lifespan.
Oh, caloric restriction. Okay.
Yeah, they did a caloric restriction study. They found it improves metabolic and immune responses that help determine both how long a person lives and how many years of good health they will enjoy while they're having this longer life. 2 years of modest calorie restriction reprogrammed the pathways in fat cells that help regulate the way mitochondria generate energy. How insane is that? That is hacking.
Yeah.
2 years of, in this case, humans. This is not a mice study. This is not a mouse study. This is a humans study. I'll repeat that. It reprogrammed the pathways in fat cells that help regulate the way mitochondria generates energy, the body's anti-inflammatory responses, and potentially the longevity of the person. This is according to Eric Ravusin, PhD Associate Director for Clinical Science at Pennington Biomedical Research Center. In other words, quotey voice here, in other words, calorie restriction rewires many of the metabolic and immune responses that boost lifespan and health span. They went further. This isn't completely correlative because they did a little bit of identifying here. As I go down here, they found, where is it? A activating factor acetyl hydrolase that was reducing. It produces health benefits that included the lower age-related inflammation and improved metabolic health. So they even found a specific thing that was taking place in the thymus. I didn't even know what a thymus is, but apparently we have one.
It makes T cells. Yes.
Oh yeah, that's where the T cells come from, is the thymus.
Yeah.
So it says here, this is quoting again Ravusin. As people age, their thymuses shrink and produce fewer T cells. As a result, older people have a harder time fighting off infections and certain cancers. Calorie restriction helps prevent the thymus from shrinking, so the person generates more T cells. They really found quite a bit of. That's important because if a person doesn't. In addition to improving immunity and increasing T cells, is associated with ability to burn stores of fatty acids for energy. And then you don't. If a person doesn't build that fuel, then you get the thing where it's building up in organs and the muscles, the livers, and the heart, and leading to insulin resistance, obesity, diabetes, all the rest of it. So it's really kind of a critical system to keep going as long as possible in its most proficient way. So there's that.
I'd like to age healthily. I'd like to, live long and prosper, right? Not have my immune system give up on me before I'm ready. What's the other thing?
This is one of those things, though, that, like Justin mentioned before, there might be something down the road you don't know, right? So there's other things with calorie restriction that you also need to look for as well.
Nope, you don't. So they did do another study. There's a second. No, you're right. There's a second study where they in mice where they reduced the calories by 40%. So they didn't do that to the humans. They got it down to 40%. And there were negative effects in growth and reproductive viability. They still got health benefits for being otherwise healthy, longevity-wise, but they had, because they had gone down so far, they had, you're right, absolutely other ramifications.
Yeah, wasn't there's also one compound, this PLA2G7, that they have identified as a potential target for people who don't necessarily want to do the caloric restriction.
Yeah,
that
so
might
if you
be a target for making the immune system stronger.
Yeah, if you just can add it to the or reducing it, I guess
Yeah.
if you could find a way to knock it out, which knocking out an enzyme is actually a somewhat easy thing. You just build a thing that it could lock into, but doesn't have any other function, and you should be able to sort of sponge up enzymes to some extent. I don't know.
Sponge the enzymes up. Yes, that sounds like amazing therapy and science. We have created the enzyme sponge. It will sponge the enzymes, and you will live forever. This is This Week in Science. I'm Dr. Kiki, and I have a couple of stories left. I hope that you're interested in hearing about cluttered old brains. A research study it's a review study in Trends in Cognitive Sciences has been looking into the idea that as you get older, you have a lot more stuff in your brain than people who are younger because you've been around for a lot longer. And so you've just got a lot more stuff rattling up in your brain. And there's this difference between wisdom and knowledge and, how can this information be used and how can the information benefit? So we also find that as we get older, memories kind of go into decline. We have a harder time accessing words, memories. Oh, remember that one time, or the name of that band, or I forgot. And you have these moments, senior moments that people talk about. Well, it could be that the brain and the memory process of older people is cluttered, but I prefer that, and this was suggested in the study, that we should call it enriched or elaborated or even overloaded.
Is that like 10 to 20 years of science news from...
Exactly. I don't even remember which story was told which week. Exactly. But what has been discovered is that as people are remembering things, younger people are much more focused in the way that they target information, and they're able to forget things that are irrelevant. They can leave things out of that memory. They don't make connections to it, and their memory stays focused on the target. And older people are better at doing this earlier in the day. But as you get older and as days go on, other memories and other networks and other things that connect conceptually from other memories of different things, they start connecting to the thing that you might be trying to remember. So the memories become, as they say, cluttered. And so what this is actually this mental process as we age. It's as people age, the brains lose the ability to focus attention on specific things. But I appreciate this study because even though older adults show reduced suppression and or deletion of information that's not relevant to a present task, they're able to make connections. This study mentions the fact that older people are able to make connections that younger people can't. And this comes back to the idea of wisdom versus just knowledge, that because of these networks, because of these supposedly irrelevant connections can be made by people who have had more memories, more thoughts, more knowledge put into their brain because there's more in there to pull from. So it's there are benefits to having a cluttered brain. Things might get lost in the shuffle. Your filing system might be a little bit difficult to get through and find things in, but the stuff is still in there, so you can get it.
So if I have a disorganized mind as a young person, is that going to make
Is it even worse as you get older? I have no idea.
Or is it just going to seem normal?
It's just, right?
Like, that's kind of like my impression is like, oh gosh, yeah, having all that clutter and all those weird connotations and things not being very organized. Wait, that's how your brain's always been, Justin. Oh, that means we'll be just fine, or it'll get worse. Ooh. Oh no.
Oh no. Yeah, so anyway, these researchers say that they argue that older and younger adult memories contain similar target features. We're remembering the same stuff, but older adults' memories contain more non-target features and so are enhanced or cluttered with excessive information.
I wonder if any of it is also because you kind of have your running task list in your brain all the time that's taking up processing power, and the older you get, the more responsibilities that you have and the more things you're constantly thinking about, and the less you can focus on the memories that you have in your brain.
So I think this is something that I don't know that anybody has really taken into account in these kinds of studies. I mean, these kinds of studies, they give you, it's in a lab situation. They say, remember this word. Don't remember this word. They give you lists of things or numbers, or you read a story and you're supposed to pull certain things out of the story. But I don't know that it does. It's always just separated by age and not necessarily by how many responsibilities you have that are potentially competing for your attention in this particular moment. Yeah.
Right. What's your current stress and anxiety level? Great.
Yeah, I think that's a good question to bring in.
I think my mind only saves annotation. Life I've lived. Got these sort of side notes in there, and that's it. Well, what was the actual thing?
Just Post-it stuck to the wall.
It kind of is. A little bit like a couple lines highlighted, something underlined. Rest.
I'm pretty sure that my brain is basically like any desk or filing cabinet that I've ever had. It's just the stuff is all still there. I haven't thrown anything out, but there's paper shoved in every crack and crevice, and it's all over the place. So,
mine is covered in doodles. So.
Doodle brain. Yeah. I love it. My last story has to do with multiple sclerosis. A study at the University of Zurich and Munich's LMU Klinikum Hospital have looked at monozygotic twins to identify the influence of environment and genetics in cases of multiple sclerosis. And in doing that, they've been able to tease apart factors that nobody has really teased apart before. So in having these monozygotic twins, which are identical twins.
That's the real twins, not the fake.
The real twins, identical
not
twins.
Not just roommates.
Yeah, not just the roommates.
These identical twins in the situation, one twin in the pair had come down with multiple sclerosis and the other had not. And so they looked to see what kind of factors conveyed the difference between the twins in order to figure out, what's going on genetically and what's going on environmentally. Because what we know is that if one of the twins got multiple sclerosis, then the genetic basis is there in both of them. So what was it that allowed for the triggering of the genes that lead to the autoimmune disorder that then the one twin suffers from? So what they were able to pull out from this is they were looking at the immune profiles, and they were able to identify proteins in the immune cells of the sick twin in each case and decode the totality of all the genes that are switched on in the cells, according to one of the researchers. And it was also added that they obtain as much information as currently technically possible from these valuable samples, and they had all algorithms and artificial intelligence to get insights from it. So what they determined is that there were hyperactive cytokine receptors. So the body's, the immune cells, were hyperactive to certain cytokines, and that led to increased activation of T cells in the blood of the patients with multiple sclerosis. And these T cells, we just talked about, they come from the thymus, but they are more likely to migrate into the central nervous system and cause damage there than other cells. Additionally, these T cells were they were naive. They were like they had been just recently activated, so they were like some kind of almost a stem cell T cell, but not quite. So they were like these baby T cells. They hadn't quite developed into fully functional T cells. So there's something odd about that particular aspect of the cytokine initiation of this particular group of T cells that go on to affect the immune system and evade the normal checks and balances. And what the researchers say is that in finding this particular group of these naive, not fully functional T cells, they say that they think they might have discovered the cellular big bang of MS here, precursor cells that give rise to disease-causing T cells, which can be very exciting. And so they will be doing further study of these twins, looking to determine more factors related to what's happening in the immune systems and the differences between the twins to figure out exactly what happens.
Yeah, it's the thing worth remembering always when we're talking about it. And a perfect example here too is that the genome is basically the building material. It's the Home Depot full of stuff that the body can use. And then things in the environment can determine sometimes what actually gets pulled off of the shelf. Yeah. And it's not a blueprint in and of itself so much as just all the stuff you're gonna get to work with. And then depends on which shelf things get pulled down from.
Previous studies recently have suggested that viruses like Epstein-Barr are highly implicated in triggering multiple sclerosis. We've even seen that SARS-CoV-2 is triggering currently some cases of multiple sclerosis. The method, like the mechanism by which that happens, why certain people genetically are predisposed and what leads to that happening. How does it now we have this new piece. Okay, there's this cytokine hyperactivity, it's something, the virus comes in and somehow it's triggering a particular thing that's making these naive T cells that cause the problem. So what is the pathway there? How is that happening? And once we start really figuring that out, we might be able to stop it.
Mm-hmm. Get those things out of there.
Cut it out, T cells.
Yeah.
Stop it. Yeah. So exciting news. Very exciting. I it's always exciting to find new answers, but we know that the complexity of the world, there are still so many questions.
I love a twin study because it removes so many confounding variables.
Yeah.
Just so many.
Yeah. Oh, you're exactly the same genetically, but you decided to do different. Oh, great. Okay.
And in many cases, you were raised in the same house, so you have the same external factors growing up. And it's great. It's scientist's dream.
How come we? Oh, I guess we do. I was going to say, how come we don't hear about how come there are a bunch of twin mouse studies? And I realize, because all of the mouse studies are twin studies.
Yeah, they come in litters. But it's even like I think about the astronauts. One went to the space station, one didn't. You got to see all the differences. It's awesome. It's It's a perfect control for so many uncontrollable factors in studying humans.
More twins. Maybe.
I don't know.
Maybe, I don't know.
There are a lot
hard.
of us here. I don't
Yeah,
know.
oh no, I'm so yeah, no. That's a terrible thing to wish
Tough.
upon
It'd
people.
be tough.
That's a tough one. Oh my goodness, I can't even imagine. One is enough. The sunk diaper fallacy.
Oh, the diapers never end. But this show is coming to an end. Thank you for joining us for another episode of This Week in Science. We're so glad that you came with us on this scientific adventure of this week. And I would really love to thank a few people. Thank you. Shout outs to Fada. Thank you so much for your help on show notes and social media. Thank you to Identity 4 for recording this week. I hope you've been having good travels. Gord and Arnlor et al., everyone who's helping to make sure that the chat rooms are nice places to be. Thank you for helping in the chat rooms. And just all of you chatters, thanks for chatting. It's really great to see you here while we're live. And Rachel, thank you for your help editing and for all of your assistance. And finally, thank you to our Patreon sponsors. Thank you to Richard Badge, Kent Northcote, Rick Loveman, Pierre Velazarb, Ralphie Figueroa, John Watnaswamy, Carl Kornfeld, Karen Tazi, Woody Yamas, Andre Bissett, Chris Wozniak, Dave Bunn, Vigard Shefstad, Hal Snyder, Jonathan Stiles, a.k.a. Don Stylo, John Lee, Ali Coffin, Maddie Perrin, Gaurav Sharma, Ragan, Don Mundis, Stephen Albaran, Daryl Myshak, Stu Pollock, Andrew Swanson, Fredus 104, Sky Luke, Paul Ronovich, Kevin Reardon, Noodles, Jack, Brian Carrington, Matt Bass, John and Nina Lamb, John McKee, Greg Riley, Mark Hessenflow, Gene Tellier, Steve Leisman, a.k.a. Z. McKen, Hayes, Howard Tan, Christopher Rabin, Dana Pearson, Richard, Brendan Minish, Johnny Gridley, Rami Day, Flying Out, Christopher Dreyer, Artyom, Greg Briggs, John Atwood, Bob Coburn, Rudy Garcia, Dave Wilkinson, Rodney Lewis, Paul, Philip Shane, Kurt Larson, Craig Landon, Mountain Sloth, Jim DePoe, Sarah Chavez, Sue Doster, Jason Olds, Dave Neighbor, Eric Knapp, E.O., Kevin Parachan, Aaron Luthen, Steve DeBell, Bob Calder, Marjorie, Paul Disney, David Summerly, Patrick Pecoraro, Tony Steele, Ulysses Adkins, and Jason Roberts. Thank you, each and every one of you, for all of your support from Patreon. And if you would like to help us out on Patreon, please head over to twist.org and click on that Patreon link. On next week's show,
We will be back 8 p.m. Pacific time on Wednesday, 5 a.m. Central European time on a Thursday.
early.
Very early. Broadcasting live from our YouTube and our Facebook channels and from twist.org/live.
Hey, do you want to listen to us as a podcast? Maybe as you pipette some goo into a dish. Just search for This Week in Science where podcasts are found. If you enjoyed the show, you can get your friends to subscribe as well.
For more information on anything you've heard here today, show notes and links to stories will be available on our website, www.twist.org.
Yes, you can also contact us directly. Email Kirsten at KirstenThisWeekinScience.com, Justin at TwistMinion@gmail.com, or me, Blair, at BlairBaz@twist.org. Just be sure to put TWIS, TWIS, in the subject line, or your email will be spam filtered into our very cluttered, or rather enriched, spam box. Yeah. We'll never see.
No, we'll never.
No.
Yeah, if you want, you can contact us directly on the Twitter, where we are at Twist Science, at Dr. Kiki, at Jackson Fly, and at Blair's Menagerie. We love your feedback. If there's a topic you would like us to cover or address, a suggestion for an interview, a haiku that comes to in the night, please let us know.
We'll be back here again next week, and we hope you'll join us for more great science news.
And if you've learned anything from the show, remember,
It's all in your head.
This Week in Science. This Week in Science. This Week in Science. This Week in Science, it's the end of the world. So I'm setting up shop, got my banner unfurled. It says the scientist is in, I'm gonna sell my advice. Show them how to stop the robots with a simple device. I'll reverse global warming with a wave of my hand. And all it'll cost you is a couple of grand. 'Cause this week's science is coming your way. So everybody listen to what I say. I use the scientific method for all that it's worth, and I'll broadcast my opinion all over the earth. 'Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in 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 Science.